Well, hello, and welcome to Life Sciences New York's Spotlight Series, a regular series of conversations with innovators, entrepreneurs, thought leaders, and folks who are driving New York's biomed and life sciences industry. Joined today by AndyBerglund, the director of the RNA Institute at the University at Albany. The RNA Institute is doing really some terrific stuff, and I'm thrilled to have Andy here with us today to talk about not only his work, but the work of the Institute in general, and give us a sense of where we're going in New York in terms of RNA research, development of products, and the like. So, Andy, thank you so much for spending some time with us today.
Thanks, Win. It's an honor to be on the show and excited to talk about RNA.
So, Andy, let's start where we like to start here at Life Sciences New York and learn a little bit about our interview subject. Yeah, I'm hoping you can talk a little bit about your past and background, what's brought you here, and uh, where we are today at the RNA Institute.
Yeah, I'll give you a little bit of a backstory. It starts when I was an undergraduate student at the University of Colorado Boulder, and I was taking a course, biophysical chemistry, and they were talking about RNA structure, and I didn't really know a lot, but I found it fascinating, and I was fortunate enough to get the opportunity to do undergraduate research. And at the time, we really didn't know a lot about RNA structure, and it was during my undergraduate research that I became sort of fascinated and fell in love with RNA. So I was able to help solve a crystal structure of RNA, and there weren't a lot known at that time. And then I went on to do my PhD in biochemistry at Brandeis University, and there I transitioned to think about something called RNA splicing. And one of the things about splicing is that we have our DNA, our blueprint, and then we make something called a message RNA that codes for the proteins. But in reality, it's not just one DNA, one RNA, one protein. What can happen is you have one gene, but it can make many versions of an RNA that codes for lots of different proteins. And so RNA splicing really gives you a lot of diversity. So, for example, we have maybe 20,000 genes in the human genome, but alternative splicing allows us to have hundreds of thousands of different versions. And so you have the same DNA in every cell, but it's all the changes in splicing that allow us to have the diversity we have within our own bodies and then across species and things like that. So RNA splicing is really important. And so I continued to be fascinated by RNA and splicing, studied that as a postdoctoral fellow. And then when I started my research as a new faculty member, now 20 plus years ago, an undergraduate here in the capital region, here in New York, introduced me to a disease called myotonic dystrophy. It's a toxic RNA disease. So unfortunately, what happens in this disease is you get it's a repeat expansion disease, which many of you may have heard of as Huntington's disease, where you have CAG repeat expansions. But in myotonic dystrophy, you have CUGs, and you have these hundreds or thousands of CUGs floating in your cells, and it's a toxic RNA. And it soaks up RNA binding proteins, and you have lots of missplicing. So understanding splicing helps us understand this form of muscular dystrophy, and that's one of my passions and a focus, I would say, one of the focuses at the RNA Institute.
And give us a little history, if you will, of the institute itself. When did it come to be? How did it come to be? And what's its purposes?
Yeah. So in the RNA Institute was founded officially in 2010, but I think it's something that's been bubbling in the capital region. So there's a lot of great RNA history in the capital region, especially at Albany, University of Albany, Department of Health, Wadsworth, for example. Joachim Frank did critical research that allowed us to understand the structure of ribosomes while he was at the Department of Health, and he was recognized with a Nobel Prize in chemistry. Recognizing that expertise that allowed us to basically see the structure of ribosomes. So those are RNA machines that make all the proteins. So again, sort of that RNA-centric world view, ribosomes. Another sort of key member that helped with this vision was Marlene Belfort, a National Academy member, who was on the leading edge of discovering RNAs that could catalyze chemical reactions. So for the longest time, RNA was thought to be this message. But in reality, RNA can catalyze chemical reactions. So RNA is pretty special because RNA can be your code, RNA can be the message to make proteins, but it can actually also fold up and do chemistry. So RNA, you could argue, can do everything. And actually, in viruses, for example, RNA is the code, it's the message. It doesn't do all the chemistry, but RNA is pretty important. And so here in Albany, they recognized that importance and decided to focus resources on the RNA Institute. So it was founded in 2010 with funding from the National Institute of Health, and New York State invested. Great institute, and it's been building for many years. I came in 2018 as the director.
And give us some sense of the research and the work that the institute's engaged in these days.
Yeah, we're lucky to have a ton of great researchers here affiliated through the University of Albany, through the Department of Health. We even have affiliates at Albany Medical Center, at RPI, and we broadly cover RNA. We think about how do you synthesize RNA? How do you make modifications so that maybe it's stable? For example, RNA modifications were key to stabilizing mRNA so it could be a vaccine, and also so you can tune the way it interacts with the immune system. You don't want to sort of have a dramatic effect on the immune system. You want to tune that effect. So RNA modifications and understanding how you modify RNA is important. But then also understanding all the RNA, also sort of the roles of RNA in the cells, in the tissues. So basically RNA biology is a big part. So RNA chemistry, RNA biology, RNA engineering. So we have faculty with engineering backgrounds that think about how can we build RNA origami, RNA structures that can do new things, new chemistry, work as scaffolds, maybe for drug delivery. So really, we go from the fundamentals of understanding RNA as a molecule to thinking about, oh, can we use RNA as a therapeutic, as a drug? And actually, what's really exciting here in New York is there's a lot of companies thinking about how to use RNA as a drug because it can really be a precise drug. You can design an RNA so that it base pairs with another RNA so that you can turn something off in the cell, turn something on. So in the last 10 years, there's been an explosion of RNA therapeutics. And that's part of that precision medicine because you can precisely say, okay, I want to turn something on or turn something off. And actually, then there's that whole area that, is sort of famous now is CRISPR. Well, CRISPR was sort of made famous because of the RNA component. So CRISPR is a protein RNA machine, and it's the RNA part of CRISPR that recognizes things in the genome and says, oh, okay, let's do some cutting. Of course, it had to be engineered, and now people are thinking about how can we fix a mutation with CRISPR RNA-based technologies? So I think it's a great time to be in the RNA world, in the RNA field, and so I'm excited to tell you about that, Win.
So, at the risk of wading into some controversy, I think, for many folks, perhaps folks not necessarily engaged day to day in the scientific community, they've heard a lot about RNA. They've heard a lot about certainly in the COVID period, right? The development of RNA based vaccines. Help ground our discussion a little bit in what does that mean to develop vaccines utilizing RNA technology, and how does that differ from, kind of the traditional view of vaccines?
Yeah. So the traditional view of vaccines is you're basically using a dead virus, right? You want the body to recognize that virus, so you mount an immune response. And so, yeah, you could use a dead RNA virus where maybe it's been mutated in some way or it's parts of it are missing. But again, it can be an RNA virus. And so for example, use COVID because that's a well-known one. What they've done there is they've just taken one part of the virus, right, the SARS-CoV-2, and they take the mRNA to code for the spike protein. And that's actually what your immune system's recognizing. And it's a way to say, okay, here, we're going to give you a little dose of the protein that your immune system's going to respond to. And it's very targeted, and the mRNA is delivered, it's an injection, and you get that mRNA, your system's going to make these proteins so your immune system can respond to it. And it's basically a minimal way of introducing your immune response. And that's what you want to do, right? You want your system to be able to recognize it. For example, if you get if you get the flu virus, if it's something your body's seen before, you do quite well. It's when it's a new virus and your body hasn't been exposed that you're in trouble. And so that's why it was important, for people to have the opportunity, if they chose to, or that may be part of the controversy that we'll not step into. But having vaccines allows your body to basically sort of be ready to put the wall up so that the virus doesn't, take over, right? It is maybe you're still going to get an infection, but your body's going to recognize it and say, oh, this is a virus. I got to fight it off, right? And so that's the real power of these vaccines. And the power of mRNA is that we can synthesize it quickly. We can turn these things around quickly so that we can respond to how the viruses are evolving, right? So that was one of the challenges in the flu, is that we were trying to sort of guess what the flu would be and say, take the flu from somewhere else, modify it. And that's why sometimes, when you get a flu shot, well, it's only going to be 30% effective. Or, they sort of were good about predicting it and it's, more like 70 or 80%. So having this mRNA technology allows us to basically, move faster and be a little more precise with our technologies. But at least to me, it's very similar technologies. You're getting part of the virus, dead virus, right? You don't want live virus. You're making the protein so that you get exposure, so that your system is exposed and will mount an immune response.
And help me understand, as we're talking about kind of uses of RNA technology in this space, are we primarily talking about the vaccine space, or what other applications are there for this technology?
Yeah, I think that the vaccine, the mRNAs are exciting, but that's just a small part of it. And actually, the Institute does a little bit in that space in terms of RNA modifications, RNA structure, understanding the RNA using computational tools. But I would say a part that's even more exciting is something called siRNAs, antisense oligonucleotides. And these are small little RNAs that can be as small as 18 to 20 nucleotides. You make them synthetically, so the chemists are involved, the medicinal chemists, and they target genes that you want to turn up or turn down. And so these are really powerful. There's now several approved drugs where they take advantage of the cellular machinery to turn on or turn off particular mRNAs. And so I think that is a really growing field. And so these small RNAs can be used therapeutically, and can be delivered in lots of different ways. So, for example, there's companies right now that are fusing an antibody fragment, for example, to an ASO. And an ASO is an antisense oligonucleotide, so it's modified RNA. And so that antibody fragment allows you to get better uptake. So, for example, into the muscles, because you're taking advantage of, say, antibody technology where there's receptors on muscle cells, for example, because maybe we want to get certain RNAs into the muscle cell. And then it gets delivered, the ASO gets released, and the ASO, back to myotonic dystrophy that I told you about earlier, recognizes this toxic RNA. And then because we've understood RNA biology and molecular biology, the ASO base pairs with the toxic RNA, and then RNase H, a little enzyme, comes along and cleaves the toxic RNA because you've introduced this modified oligonucleotide, antisense oligonucleotide. So that's a huge industry. So many companies. I mean, I can think of just 50 that I know are using this technology, ASO siRNAs, and it's powerful. You can, any type of genetic disease people are using. You can also use them to target viruses, to degrade viruses and things like that. So lots of technologies that people are using with RNAs. And the beautiful thing about RNA is it tends to be transient. It's, you're going to turn things on or off, but you can stop using it if there's issues. I think one of the challenges we're going to need to overcome is that, CRISPR technologies are really exciting, really powerful, but if you're changing your DNA, changing DNA back is hard, right? When you're making wholesale DNA changes, that's a change that's probably permanent, right? But if you're using RNA as a therapeutic or drug, you can go in and you can use it just for, a few days or longer, depending on how you want to do it, but you have a little more potential control with RNA drugs.
And you've talked about applications in the virus space. You've talked about applications in muscular dystrophy, I presume other neurodegenerative conditions. Give me a sense of just how broad the potential applications are in this space. Are we limited to certain disease states, or kind of give me a sense of where you think this work is going to go over the next 5, 10, 15 years?
Yeah, I think there's a lot of opportunities. I'm sure there's certain diseases that maybe you couldn't, aren't easy to target with RNA, but it's every time I go to a new RNA meeting that has a therapeutic component, I'm excited to hear about sort of new strategies they're considering. Like things you can do is you can identify RNAs through something called in vitro selection, where you literally can sample 10 to the 14th different RNAs, just like sort of developing an antibody. You can develop an RNA to recognize sort of any cellular target you want, and then you could use that to selectively bind a protein or a small molecule and modulate things. So RNA has in a way, I think about it as just another tool in the um, drug discovery pipeline, right? You can use antibodies, you can use small molecules, those are still really powerful. But now RNA, I would say, is a really important drug modality that people are using. Almost all the big pharma, you know, Pfizer, Regeneron, Merck, Novartis, they all have an RNA effort. It may not have anything to do with mRNA. It may have to do with using small RNAs, these siRNAs or ASOs, so that they can target a gene and modulate it. So, it's a big industry. And actually, that's one of the things that we're wanting to help support is that we do a lot of the training of the next generation of RNA scientists. Actually, that was one of the things when I became director of the RNA Institute. I said, our mission should be to train the next generation of RNA researchers. And so that I'm really passionate about. I've been doing that for many years. And that's actually part of why, we were excited to get a center of excellence in RNA research and therapeutics so that we could marshal resources from the state to really push that mission forward. And we do that in collaboration with the University of Rochester, who have outstanding researchers in their Center for RNA Biology. So the RNA Institute here at the University of Albany, the Center for RNA Biology at the University of Rochester, we've come together in a New York State partnership and funded by the state to do this work on the Center of Excellence in RNA Research and Therapeutics. So we're helping support startups. We help support larger industry. We want to support everyone who wants to do RNA research in New York. Of course, we'll support those outside of New York, but we're New York centric, of course.
And for folks who are interested in learning more about the RNA Institute, learning about ways that they might connect and collaborate, what's the best way to do that, Andy?
Yeah, so you can come to our website, and there's lots of ways to engage with us. I think we have literally rna@albany.edu is an email that will reach my staff and myself, and you can engage with us. We hold meetings that are part of the Center of Excellence in RNA Research and Therapeutics jointly with Rochester. We've held them at Lake Canandaigua. We hold them here at the University of Albany, and that's going to be a yearly meeting where it's an opportunity to engage with industry. And we're also open to lots of partnerships. As you know, Win, we're going to partner with you at Life Sciences New York in November this year. And that's an opportunity to chat with us and talk about collaborations.
Well, Andy Berglund, I appreciate you spending time. Unfortunately, these conversations always seem to fly by too fast, but I appreciate you spending some time with us. For folks who are interested in connecting with Andy and his team, we will have those websites in the chat below. But Andy Berglund, director of the RNA Institute at University of Albany, thank you so much for spending some time with us today, and we'll keep talking.
Thanks, Win. Nice conversation.